The learning objectives for this lecture are to understand the function, location, and regulation of lipogenesis, as well as some special features. You need to know the steps of synthesis from glucose, as well as the synthesis from a special case with acetate. You need to be able to integrate the fat biosynthesis, or lipogenesis, growing of fats, with fat catabolism, and explain the overall metabolism of adipose tissue. Okay, so the catabolic and the anabolic processes. So the synthesis of fat from glucose is essentially making fat. So it's putting lots of carbons and hydrogens in a string together. So much like beta oxidation was breaking these down, 2 carbons at a time, this is the process of building them up. The scenario goes up to, or lipogenesis goes up to C16. So it's putting 16 of these carbons and hydrogens together. To do this, we need a source. We need a source of carbon. That source of carbon is acetyl CoA. We also need a source of hydrogens. Okay, so hydrogens or electrons, and that's in the form of NADPH. But where does this occur? Well, as you can see from the table below, lipogenesis occurs in different locations or in different tissues for different species. For pigs, it does not occur in the liver, as well as ruminants. That does not occur in the liver, but in the adipose. For rats, mice, and rabbits, it occurs in both the liver and the adipose. Whereas humans and birds, it only occurs in the liver, not the adipose cell. The location of lipogenesis at a cellular level is also very important. It occurs in the cytoplasm, but we need a source of carbon, as mentioned before. That source is acetyl CoA. We also need a source of hydrogen in the form of NADPH. And a number of these substrates are actually generated in the mitochondrial matrix, so they're not readily available in the cytoplasm. So this brings us to the steps of lipogenesis. As mentioned before, acetyl CoA is mostly formed in the mitochondria. So the first step would be the formation of acetyl CoA into the cytoplasm. This is performed using a citrate cleavage pathway, which we were going to go through shortly. We also need to control the rate limiting step, which is acetyl CoA carboxylase. This enzyme actually makes malonyl CoA, as we've mentioned before in beta oxidation, is the first step of the synthesis of fatty acids. Then we have the next step of fatty acid synthase. This actually makes fatty acids 2 carbons at a time using acetyl CoA. And then finally, we need a provision of reducing power through NADPH. And this can be supplied 50% through the citrate cleavage pathway, as well as 50% from the pentose phosphate pathway. So step 1, the formation of acetyl CoA in the cytoplasm, which is essentially the citrate cleavage pathway. So this is the full cycle through cytoplasm, mitochondria, but I'm just going to start stepping you through from glucose. So from glucose, we can go through glycolysis to pyruvate. Okay, pyruvate can now penetrate from the cytoplasm into the mitochondria, much to the scenario through the TCA cycle. From there, pyruvate through pyruvate carboxylase can go to oxaloacetate, citrate synthase, with an input of acetyl CoA and ATP as we go. Pyruvate, obviously through pyruvate dehydrogenase, also produces acetyl CoA. It can be formed from multiple sources. So we now have acetyl CoA in the mitochondria, but we need that in the cytoplasm. That's where the citrate comes through. So citrate can actually flux through this mitochondrial wall, okay, and be formed in the cytoplasm. From there, we have an enzyme, ATP citrate lyase, which is in the cytoplasm, which then converts citrate back to oxaloacetate and acetyl CoA with an input of ATP. Oxaloacetate has an input of NADH to malate, and then malate back to pyruvate using NADP malate dehydrogenase producing NADPH. The thing is, we've now got cytoplasmic NADPH, and we've also got acetyl CoA in the cytoplasm. So these are the building blocks we need to produce fatty acids through fatty acid synthesis. So the function of the citrate cleavage pathway is the formation of acetyl CoA in the cytoplasm, and that's through the enzyme ATP citrate lyase. So ATP requires some ATP. The formation, it also is a function of the formation of NADPH for fatty acid synthesis in the cytoplasm, and that's through the enzyme NADP malate dehydrogenase. Okay, there's a regulation steps where insulin actually stimulates the gene transcription for both of these enzymes. So ATP citrate lyase and NADP malate dehydrogenase. Insulin actually upregulates the transcription. So in reality, a high carbohydrate diet, which is resulting in insulin being released, will actually upregulate fatty acid synthesis. So as you can see here, insulin is actually up transcribing these two enzymes. So if you will, high glucose in the diet is resulting in increased fatty acid synthesis because of the upregulation of these genes. Step 2, acetyl CoA carboxylase is the key rate limiting step. So this is that formation of malonyl CoA. So let's just have a look at the whole cycle altogether. And let's just focus here on this area of citrate to acetyl CoA into fatty acid synthesis. So we've got this citrate and acetyl CoA now in the cytoplasm, and now we need to form malonyl CoA. That's through the catalyzed reaction of acetyl CoA carboxylase. Okay, it uses bicarbonate coming in, and we're using ATP as well to form malonyl CoA. The function is the formation of malonyl CoA, and it supplies 2 carbon units for fatty acid synthesis. Like all carboxylases, it contains a biotin as a prosthetic group, and this is the rate limiting step, which means it has some regulation processes. So let's have a look at the regulation. So firstly, it is regulated through the cyclic AMP mediated reversible phosphorylation, and it is active in the dephosphorylated form, which means insulin reduces cyclic AMP and thus the dephospho form, and so it activates. So insulin stimulates the gene to be transcribed and produce more of this enzyme. So in reality, looking at insulin usually means that you're in a fed state. So more fed, we don't need to oxidize fats, we need to build them and store them. In the scenario of glucagon and adrenaline, it actually increases the cyclic AMP stimulation. And so this is in the phosphorylated form, so it actually inhibits the enzyme. So this enzyme is now inhibited as it is phosphorylated. There's also allosteric inhibition or regulation here. So the more NEFA-CoA that you have, it actually inhibits the enzyme. So if we've got too much NEFA-CoA or too many fatty acids, we actually inhibit. If we've got a lot of citrate formed in the cytoplasm, that also allosterically activates the acetyl CoA carboxylase. Okay, so once this citrate cleavage pathway has popped citrate into the cytoplasm, it will most likely flow through acetyl CoA to NEFA-CoA. So remember, there's also that regulation step here of malonyl CoA. It actually speaks to beta oxidation. So remember, malonyl CoA actually inhibits the formation of this carnitine binding to the NEFA-CoA to form NEFA-carnitine, which then stops or blocks NEFA from entering into the mitochondrial matrix. So remember, carnitine palmitoyltransferase, CPT1, is inhibited by malonyl CoA. So if you like, beta oxidation and lipogenesis talk to each other. So when synthesizing long-chain fatty acids in the well-fed state, Acetyl CoA or the NEFA-CoA will not bind to carnitine, and thus there will be no substrate for beta oxidation. Step 3, fatty acid synthase. So fatty acid synthase, its function is really to build that NEFA-CoA. It is a complex of enzymes that makes fatty acids through a series of steps of reduction, dehydration, and reduction. It adds 2 carbons at a time, which is supplied from malonyl CoA, which again, remember, is coming from acetyl CoA, which is coming from the citrate cleavage pathway. It needs hydrogen ions or electrons, which is supplied through NADPH. Okay, that again is supplied from citrate cleavage pathway and 50% from the pentose phosphate pathway. The end product is C16 or palmitate. The regulation of this, essentially, insulin stimulates the gene to be transcribed and produces more of this enzyme. So in the well-fed scenario, we're producing more or transcribing more fatty acid synthase. So we're really getting more fatty acid synthesis. Step 4, provision of reducing power. So as mentioned before, the provision of the reducing power or the hydrogens or the electrons, 50% comes from the citrate cleavage pathway through that reaction catalyzed by NADP malate dehydrogenase. The other 50% comes from the pentose phosphate pathway, i.e., glucose 6-phosphate dehydrogenase. Can anyone think of the other function of the pentose phosphate pathway? Have a think. So now I'm going to step you through the special scenario of lipogenesis from acetate in comparison to what we went through with glucose. Lipogenesis from acetate. When does it occur? Well, in ruminants, acetate is the main source of carbon, and that's required for lipogenesis. So ruminants typically do not absorb glucose, as the diet it is fermented to volatile fatty acids. And we'll learn a little bit more about this in the ruminant lectures. Hence, glucose is actually spared and acetate is used instead. In the scenario of humans, we can actually use acetate as a form to store fats or lipogenesis when we consume ethanol. So ethanol is converted to acetate in the liver, and we can actually then produce fatty acid. So essentially, if you're drinking a lot of beer and have a high-fat diet, you're going to end up looking like this guy. So overall, lipogenesis from acetate is much similar because you can actually generate acetyl CoA in the cytoplasm quite simply through acetate. However, we still need to get that reducing power of NADPH. So you can also send acetate back through into the mitochondria, back through the citrate cleavage pathway to produce NADPH. The other 50% will be again supplied by the pentose phosphate pathway. So lipogenesis from acetate in ruminants has a special feature, and that is NADP isocitrate dehydrogenase. Since acetate is readily forming acetyl CoA in the cytoplasm, there is not necessarily any need for acetate to enter the mitochondria. Thus, there's less reliance on the citrate cleavage pathway to provide acetyl CoA in the cytoplasm. And there is absolutely less dependence on glucose to form this acetyl CoA. Therefore, most of the NADPH actually comes from NADP isocitrate dehydrogenase, as you can see here, generating alpha-ketoglutarate, which can then flux back through into the mitochondria. So 50% of that NADP, or that reducing power, is provided through NADP isocitrate dehydrogenase, which is a special form in the cytoplasm. The rest is the pentose phosphate pathway, again, much like in non-ruminant animals. So going back through our objectives, you've gone through the function, location, regulation, and special features of lipogenesis. You've gone through the steps for the synthesis from glucose, and also looked at the special case of synthesis from acetate. So now we're going to run you through a bit of an overview of fatty acid metabolism. As you can see, from triacylglycerolysis storage, lipolysis, running down to beta oxidation, or building and esterification and storage, they kind of talk to each other. So in the fed state, we have high carbohydrates or glucose coming in, amino acids forming acetyl CoA. We also have high dietary fats. In this scenario with carbohydrates, acetyl CoA forming lipogenesis and storage of fats, or building the biosynthesis. So from there you have esterification to triacylglycerols. This dietary fat building in the liver results in this fatty acid. These fatty acids can then be esterified and taken along in very low density lipoproteins to the extrahepatic tissues such as the liver and the muscle. In these scenarios, you have extra glucose, and therefore you have insulin release. Therefore you have the upregulation or promoting of GLUT4 receptors. So the muscle is actually going to take up a little bit more of this glucose as well. So you're not going to have that full storage of lipogenesis. You also have insulin increasing, or glucose releasing insulin, and also increasing the lipoprotein lipase activity in the adipose. So therefore, that triacylglycerol that's stored or transported on those very low density lipoproteins is actually being uptaken by the adipose tissue for storage a little bit more. Remember the km value for the muscle in the lipoprotein lipase on the capillaries? It's pretty low, and so it's actually just turning over a good substrate concentration. In this scenario where we've got dietary fats coming in, and they're coming in as triacylglycerols already, they're transported on chylomicrons, okay? And they are delivered to the adipose and the muscle extrahepatic tissues. In this scenario where our diet is mostly fats coming in as triacylglycerols, if they're free fatty acids, the esterification, from that triacylglycerols, you have most of your energy coming from beta oxidation, okay, going and producing energy through acetyl CoA. If you've got a lot of pressure from that in the fasted state, you have the release of glucagon, which sets off the cyclic AMP system, turns on hormone-sensitive lipase, which in the adipose tissue releases lipolysis, which cleaves off the glycerol to produce free fatty acid, non-esterified fatty acid. This can be transferred on serum albumin, the fatty acids after lipolysis, transported to the liver for beta oxidation, okay, which produces acetyl CoA and a bunch of other FADs and NADs for energy. Acetyl CoA can transport into the TCA cycle. Okay, this can be transported to or done and produced in the liver and extrahepatic tissues as well. In a scenario where we have a lot of glucagon and a lot of fasting, you start producing ketogenesis or ketone bodies. This occurs in the liver, and it spares this TCA cycle, kind of slows it down, and produces these ketone bodies. Under extreme glucose pressure, with a lot of glucon, extreme fasting, altered physiological states, you have a lot more lipolysis occurring. So lipolysis then leads into fatty acid, beta oxidation, to acetyl CoAs, goes to ketogenesis, and you're producing a lot of ketone bodies. So you're removing a lot of the non-esterified fatty acids in the blood, but you're replacing it with ketone bodies. And the more of that, you can get ketoacidosis, rising of the acidity in the blood. This figure here shows the relative changes in blood parameters during fasting. So this is the level and this is the hours of fasting. Firstly, the scenario of fasting where our blood glucose were continually being monitored and kept at bay by regular hormones, just staying at bay. Once the blood glucose starts dropping because we've got no supply from our diet, our glucagon will go up while our insulin will go down. At the same time, our liver glycogen is slowly being used and depleted to maintain this blood glucose level. The addition of glucagon has allowed for hormone-sensitive lipase to start utilizing and mobilizing some of our fatty acid stores from our adipose tissue. So our free plasma or free fatty acids is increasing, and our liver takes it up and starts using that as a source of energy. At the same time, when the fatty acids get a lot higher, it starts switching to ketosis or producing ketones in the blood to actually start reducing some of that free fatty acid plasma. So the further we go, the more ketone bodies we get, the less glycogen. And by the time we get to this 24 hours, it's probably completely switched to producing or metabolizing through the fat mobilization. So that leaves us today, and up next is going to be the essential fatty acids lectures. Thanks again.